Hi, this is Mr. Smalley. Thanks for tuning in to this video tutorial, and what I want to talk to you about today is this question of what is science? So that's actually more interesting of a question than it appears at first light. So if we have a person here, and let's say she wants to ask a question, okay? So she's asking a question about something. Now, it actually matters what you ask a question about in science. Science really can only answer those questions which are going to look at things in the universe. That would include our world and everything in it. It also is going to look at things that are natural. Natural things, the way I would define that is going to be the physical things of the universe and energy. Now, I would contrast that with things that are supernatural. This prefix here, super, actually means above. So by definition, supernatural things are above or beyond measurement, okay? So it's sort of outside of the realm of what we can measure. So these things are going to be studied using different sources of or epistemologies, ways of coming to knowledge, and they have to be accepted oftentimes on faith. For instance, something that would be supernatural would be love. So I could say something like, I love my wife, which I do, but I could never measure or quantify that love. So it would have to be accepted just by belief. And science, when we're looking at the natural world, one thing that's nice about science is we can actually be skeptical about things. And we can question things that don't have valid justification. So an example of that is oftentimes people will say why things are occurring. For instance, the ulcers were thought to be caused for a long time by stress. And then there really wasn't incredible good amounts of justification for that. But later, when people became skeptical about that claim, they actually found out that different types of bacteria were really the source of what was causing ulcers in people's stomachs. Another important term to know about science is empirical. Empirical is going to be something that's measurable, repeatable, and observable by many people, and it's not based on personal bias. Bias is a prejudice or a scenario where somebody has already framed in the way that they think something works. And this can be due to many different things. It might be their own cultural bias, their personal history that leads them to have a prejudice, or even money, which is often involved in research, can really skew or throw off the conclusions that scientists have about their thing that they're studying. Now, I also want to address a scientific theory. This is a lot different than theories that you come across in everyday language. Theory in science is going to be a system of ideas that explains many observations and is supported by a large body of evidence. So the way that I sort of think about this visually is that there's many columns or pillars of support. So for instance, in my discipline, biology, the most important theory is the theory of evolution. And that's supported by many different lines of evidence. So we could talk about things like natural selection, or in this case, this is a picture of a Chihuahua skull and a Great Dane skull. And this is a great example of artificial selection. And artificial selection basically means that we started out with one common ancestor that would have been a wolf, and that wolf was able to give rise to two very not similar offspring over just a short number of generations, being the Chihuahua and the Great Dane. Another example would be something like natural selection. So you can see that in certain finches on the Galapagos Islands, their beaks changed from generation to generation. The beaks, for one generation, they will be really well suited to well, it depends on the offspring. They could be really well suited to whatever environmental conditions there are, and those environmental conditions could benefit that individual, and if they benefit them, they can pass on their genes. If they don't benefit them, that can actually lead to their death, and so they don't get to pass on their genes at all. Another line of evidence would be something like the fossil record. So this example is of horses. If you dig deeper and deeper back into layers of rock, you can actually go further and further back in time and look at the forms that these horses' bodies had. And you can actually find things like as time goes on, their body size gets larger, and actually old horses used to have different numbers of toes, and so they're changing over time. Finally, we can look at evidence from things like genetics. These are a series of genes called Hox genes. Hox genes are basically the body layout plans for different animals, and it's really interesting when we look at these genes. They're the same, essentially the same information for how to do the body plan for things that are like a fly or a worm, all the way up to more complex organisms like humans and mice. So the important thing to note here is that a scientific theory, okay, this is not just a guess. Okay, it's actually based on strong lines of evidence that have been tested, they've been observed, and they're repeatable. Okay, so I said earlier that science is empirical. That means it's based on measurement, and in science we use the metric system. I come from America, and unfortunately we use different units of measurement than the metric system. So the important ones to note would be liters for volume, grams for mass, meters would be for length. What's nice about this is if we have something, we'll just say we have 44.0 grams of gold. So if we wanted to figure out, how many kilograms of gold we had, we would simply be able to divide by 10 for each step that we go up here, and we could do that but by simply moving the decimal point. So I'll do that real quickly here. I'm going to move it up 1, 2, 3 spots. So 1, 2, 3. And I'm going to rewrite it up here. The way I would write that now is 0.044 kilograms. Okay, so the way I'd read that is 0.044 kilograms. So we simply are moving up and down on this staircase of metric conversions. The other type of data that we often use in science is called qualitative data. Qualitative data records descriptive observations. So if I was going to look at something about the gold, I could say, it's interesting how the pure gold, I can actually bend it. It's malleable. Gold has a gold color. It has the ability to reflect light. Those are descriptive observations that we can't really quantify or put into numbers, but they're often important in science. Okay, let's talk about the components of an experiment. So I want to tell you about the first experiment that I ever ran with any of my students. When I was a young teacher, I went into Walmart one night and it was midnight. And as I was walking the halls of Walmart very late at night, I came across these incredible 25-cent ghost shrimp. So these are ghost shrimps right here. And so I bought some of them, just spur of the moment. They were really cheap, but nobody at the store could tell me what they ate. And so I just had this huge question: what do these eat? Okay. Now I had a simple clue, which was that there was a lot of algae in one of these stomachs, and the reason I thought that was I could see a little green spot. So I came up with this hypothesis, which is just simply a prediction that can be tested by an experiment. So my hypothesis was these things are eating algae. Okay, and I could write that out in a number of different ways. I like to stick to just a really open way of writing a hypothesis that's clearly stated prediction that we can test with an experiment. Now, in our experiment, there's two variables I want to mention right now. The first is the independent variable. So this is going to be a factor that an experimenter changes in an experiment. And that's actually different than the dependent variable, which is the factor that changes in an experiment. Let me give you one tip for how to remember that. The independent variable, this is what I change. And the dependent variable, this is going to be what data I collect. Okay, so D is for data. I is for independent variable is going to be what we have for I, D for dependent variable. The final thing I want to talk about is a control group. A control group serves as a standard of comparison because the group receives no experimental treatment. So I'll show you an example of that in this next experiment. There's one more variable that I do want to mention here. That's going to be a control variable. So a control variable, this is basically going to mean that we have to keep all the conditions the same except for the independent variable. So this should be the one thing that we change. So this is the experiment I ran. I got quite a few of these shrimp here. These are ghost shrimp. And I put these ghost shrimp into 10-gallon aquariums, and I put a quadrant into each one of these aquariums, and I put in different types of food. So over here I had algae, here I had fish flakes, and here I had chicken liver. Now what I did was I put them in here, and every 30 seconds I counted where the shrimp were and how many were in that corner. So there might have been one in this corner after 30 seconds. There might have been 4 in this corner. There might have been, let's see here, 1 in this corner and maybe 1 in this corner. So what I was going to do is I was going to see where they spent most of their time, and based off of where they spent most of their time, I assumed that they would be eating in those quadrants. So my independent variable would have been the type of food that I put in each quadrant. My dependent variable was what I measured, which was the number of ghost shrimp in each quadrant. The control variable, I actually ran this many different times, and so I wanted to make sure that everything was the same. So I had the same type of shrimp, the same type of algae, same type of fish flakes, and same temperature, same type of chicken liver. Everything was the same except for that one variable that I changed, which was the type of food. Okay, so the last thing I want to look at here is graphing. Graphing is a really cool way to visually represent what your findings are in any experiment. So you want to start out with a title that describes what you were trying to study. In this case, it was shrimp food preference. And on your y-axis, you're going to want to put in my class. I always try and put the dependent variable over here. This is not essential, and many good graphs are made without doing the dependent variable on the y-axis. However, it's sort of a standard way that it works, and so oftentimes I think it's helpful to just set it up that way. On our x-axis, we're going to put our independent variable, independent v. This is our x-axis over here, okay, running horizontally across the bottom. We also have a legend here. This is not a very full fairy tale. It's actually basically just something that helps us identify what each of these different colors are, and it labels everything that we need to have labeled on our graph. Now, I want to include an important thing here. So my data for my experiment showed that the shrimp were preferring to spend most of their time in the chamber where there was fish food. So the conclusion of my experiment was that the shrimp preferred fish food, and so therefore I continued to feed them fish food throughout the year. Now, I do want to talk about this. When I look at this experiment, oftentimes people say that I proved the shrimp eat only fish flakes. I actually want to move away from that. So I didn't exactly prove this. What I did is I showed that it's statistically very likely. Okay, so the way I would say this is it's very likely it's very likely that these shrimp actually did prefer the fish food, but we never want to say that something's proven, because later on somebody could come across and do the exact same experiment, and they might find different results, and those results might be even more conclusive about what the shrimp actually prefer. So thank you so much for tuning in, and I hope that you've learned something.